Hello everybody. Today I'd like to talk to you again about natural selection as it regards antibiotic resistance in bacteria. So natural selection, as you can imagine if you've been paying attention to the lectures and the different concepts in the chapter, is a really important thing that we have to pay attention to in the environment, as well as just having to do with our medical use of antibiotics and many other things. So we looked at natural selection as regards insecticides earlier in a previous chapter, and it was brought up again in this chapter about how insects, when they're exposed at high levels to insecticides, will naturally gain resistance to them because it's a natural process of evolution to be constantly adapting to your environment. And if you do have the genes that are able to survive a given environmental pressure, like an insecticide or an antibiotic, then you're going to survive and you're going to pass on those successful genes to the next generation. Whereas if you don't have those successful genes, you're going to die. So we see that example here again with antibiotic resistance. So I'd like to bring it up again since it is such a really important concept in all of biology as well as environmental science. But let's talk specifically about how this relates to antibiotic use. So we could be talking here about humans or cattle, as we saw discussed in the book. It applies to everything, but let's look at what's going on here. So here you have a whole bunch of bacteria. Let's say it's on a petri dish that we're looking at in the lab. This, of course, could be out in real life as well. So we have two types of bacteria that are growing. They're the same species. Some of them, however, have genes that allow them to be resistant to a given antibiotic, and others do not have the genes that allow them to be resistant to that antibiotic. So if the particular antibiotic we're interested in that some of them are resistant to would be this one here, say penicillin, for example, and we have this population of bacteria. They're all the same species. They have almost entirely the same genetic components, except for maybe a couple of things, and one of them may be this resistance to the particular antibiotic. Here's our population. Now all of a sudden the whole population gets exposed to the antibiotic at the same time. Well, obviously those that just genetically have the advantage of being able to survive that particular antibiotic, and there's lots of reasons for that I won't get into in detail now, but you can ask me about it if you're interested. Those that have the antibiotic resistance naturally are obviously going to survive the pressure of being exposed to that antibiotic. Maybe some of these others will survive just because of their location or the dose of the antibiotic. Maybe they're on the fringes and they're not exposed to maybe as much as those bacteria in the center here. For whatever reason, we have a certain population now that's going to be able to survive that antibiotic exposure. Therefore, any of these that died in response to the antibiotic, their genes die with them, and their potential antibiotic resistance dies with them. The ones that survive are the ones that get to mate if they're sexual organisms, which bacteria are not usually, but these guys are going to divide, literally duplicate themselves and divide in half. So if they have the surviving gene that's important for surviving that antibiotic, then their progeny will all have the same gene that helps them survive that antibiotic as well. And as this population grows, we will see more and more of the antibiotic-resistant colored bacteria just because those are the ones that are surviving, and those are the genes that are being passed on. Same thing we saw with insecticides earlier. Now, if we come back with the same exact antibiotic and expose the second population, which has already survived the first round, if we expose the second population to the same antibiotic again, then you would expect, again, because we have more antibiotic resistance in this population than we did in the first one, we would expect more and more of them to be able to survive and hand on their resistance genes to the next generation. So how is this important to what we're talking about? Well, whether you're talking about humans or cattle being exposed to antibiotic, the important thing to remember is the more antibiotic that individual is exposed to, and the more times it's exposed to it, the more the possibility that they will be harboring a population of resistant organisms in their body. So if you're a human and you've taken antibiotic A one time and you've taken it appropriately, there's a chance that some of the bacteria that you had that were making you sick could be resistant to that antibiotic. And if they're able to stick around for a while and some later point in time you're going to need to take that antibiotic again, you're just increasing the resistance to that antibiotic every single time you expose the bacteria population to that antibiotic. So this is how we get resistance over time. And if we're doing this on a large scale with a whole population of humans or of cattle, you can imagine that in a very short period of time, if we have overexposure to antibiotics, we could have increased resistance to that antibiotic and indeed get to the point where that antibiotic is really no longer useful to us. When we stop and think about the fact that as humans, our major threat to our survival is our diseases. We don't have a lot of other organisms out there hunting us and killing us besides each other. Our major threat is diseases, and antibiotics are one of the only thing we have to be able to protect us from those diseases. We definitely want to keep these medicines as safe and as resistance-free as long as we can. This can be applied to cattle as well and other livestock like pigs and things. So especially in the United States, we've been feeding our cattle antibiotics just in their water and their feed to prevent, kind of as a prophylaxis, to prevent any kind of disease from happening. So that, of course, we can get better returns on our investment, we can get more meat out from each cattle, and we have less die-off. However, this is a problem because if you're constantly feeding antibiotics to the cattle, then obviously you're building up resistance in those cattle over time. And guess what? These are the same antibiotics that we use as humans. So we're building up resistant bacteria populations in these cattle, which can be somewhat easily transferred to the humans that take care of them and transferred into the population. So that now we have a new superbug that's resistant to antibiotics that we're going to have a really hard time treating if the humans get sick with it. Another thing that we see in this picture is one of the major reasons we give antibiotics is not just to increase our profit, but so that we can have this kind of crowding environment and we can have more animals in a smaller space getting less sick because we're treating them. But this provides, obviously, unsanitary conditions, increases the chances of them getting sick, and then when we have the resistant bugs going on, can be even a greater problem in the end. Here we have kind of a comparison of what antibiotics are being used for as far as farming goes. So the orange line here represents the antibiotics sold for meat and poultry production versus the yellow, which has to do with treating humans. So you can see that relatively stable line of human treatment over time with antibiotics. This is from 2001 to 2011. So in this 10-year period, we were pretty stable with our treatment of humans as far as antibiotics go, but we've put an increase since 2003, a pretty dramatic increase in the number of animals that have been treated with antibiotics. That's just not because animals are getting sicker, but because of the crowding conditions and because of the fact that we're treating them prophylactically. So we're using our antibiotics very inappropriately when we're using them on animals. This will make them less effective overall for humans in the end. Another figure from your book here talks about, well, what would happen if we just kind of actually start to decrease the use of antibiotics, for example, macrolides, which is a type of antibiotic, which is represented by this blue line here. So over time, we have decreased the consumption of antibiotic in, I believe this was pigs. If we look back at the book, we've decreased the use of antibiotics in pigs as prophylactics and only been treating them when they're sick, and therefore we've given them more space to move around and clean their pens out better to try to prevent the normal diseases that would happen. So we've really reduced the macrolide use. Here, this is in, I believe, Denmark. But what happens then to the amount of type of bacteria? This is C. coli. That is a typical bacteria in those pigs that we would find. And here you see that we've also had a decrease in the amount of C. coli cases. So if you stop focusing on trying to prevent the disease and actually provide conditions which would prevent the disease, more space for the pigs to roam around, cleaner environments, cleaned out, so they're not interacting with each other and they're not interacting with their own feces and each other's feces and things like that so much while they're eating, then you can see that just taking basic environmental type of preventative measures for preventing the bacteria have decreased the bacteria and have allowed the people in Denmark to use less antibiotics overall. So if we can understand how the environmental situation works in our cattle and our livestock and all the other types of animals that we may be raising, we don't have to be using these macrolides, which are perfectly good to be used on humans and increasing resistance in these livestock.